right, in chapter 3, we are going to talk about what we call the biological macromolecules. Remember, macro means large, macromolecules are big molecules. So these are the 4 primary types of molecules that are found in biological systems. We're going to build on our little bit of chemistry that we learned, and we will talk a little bit about some of the bonds that are involved in here. But remember that our main takeaway here is that any sort of bond is stored energy. When you break the bond, you release energy. It takes energy to make the bond. So these are the types of molecules that make up most of life. You've probably heard of some of these: carbohydrates, lipids. You might not have heard of nucleic acids and that terminology, but you've probably heard of DNA and RNA. You'll notice I'm going a little bit out of order from the book because genetically and in the body, it actually makes sense to talk about the nucleic acids first and then this thing called proteins here. But we're going to start with some basic general stuff about macromolecules because they tend to be made up of small units stuck together. So let's talk about them. Okay, so we're going to talk about ways that biological macromolecules get made and broken down. So to do that, we have to talk about 2 types of general reactions. These are not specific, like here we have glucose and glucose being added together. This is a reaction, but we could have 2 other sugars, fructose and glucose, or something like that being added together. And the same kind of process is going to occur here. So we're going to talk about how macromolecules get made, and then we'll talk a little bit about how they get broken down. All right, so a biological macromolecule is a large molecule that's necessary for life that is built from smaller organic molecules. Here you can see 1 unit of glucose plus another unit of glucose. We have this reaction that occurs, and they get stuck together, and they form a new, what we call disaccharide, called maltose. That process, broadly happening, is called polymerization. We take 2 what we call monomers and we stick them together and we get a dimer. And if we add more monomers to that, we get a polymer. So a monomer is just any small unit, and then when we link these monomers together, we build a polymer of this. The monomer can be different. For the different macromolecules, there are different monomers, but we will talk a lot about this in DNA because DNA is a very, very long polymer. Proteins are also polymers of amino acids. So we'll talk about these, but we're going to start with sugars because this might be most familiar to you. So let's take something like glucose, C6H12O6. That's the formula for glucose. It can form these ring-like structures. Glucose is a simple sugar. You might be aware of this if you're into exercise science and stuff like that. Glucose, we break down sugars into glucose so we can utilize them. We will talk about all the chemical reactions that do this in this course. It's really cool because you break down glucose almost identically to how bacteria do because of your shared evolutionary lineage. The reaction really hasn't changed that much over those whatever billion years that you've evolved differently from them. But we can take glucose monomers and stick them together and build something called starch. Starches are polymers that include somewhere around 300 to 600 glucose monomers being stuck together. So you can see here's 1. Then we have 300 to 600 here in the middle. This would be much longer, obviously. And then 1 at the end here. So a starch is a big chain of glucose molecules. Okay, so let's think about our own nutrition, right? You probably are aware that starch-rich foods have lots of energy in them. That's because you can take each one of these glucose molecules and cut it off and send it into a process in your body and break it down for energy. And there's 300 of these. So 1 long molecule of starch is going to have 300 times as much energy as 1 small molecule of glucose because of that. Okay, so that's the basics here of how macromolecules are built. We'll talk now about the 4 types, and then we'll define the reaction that goes into building these things. All right, so you have your carbohydrates first. We're going to talk about These are sugars. They can be used as food sources and energy sources, but there are also some carbohydrates that are structural components of cells. Let's think about plants. Plants have a cell wall made out of cellulose, and that cellulose is a carbohydrate. It is very tough and rigid and allows plants to stand upright. There are some other structural carbohydrates like chitin and things like that that are involved in cell walls of like fungi and in some animal structures. We then have lipids. Lipids are very useful for energy storage, but also lipids make up a large part of our cellular membrane. We're going to talk all about membranes because we're going to talk about cells, but lipids are very important. These include fats and oils. There are some lipids that are also converted into animal hormones that are lipid-based. That's why some like steroid or hormonal creams, you don't want to get them on your skin unless you're supposed to be taking them because they can be absorbed through the cell membrane, which is made of lipids. We'll talk more about that as we get closer to that. We'll talk about hormones a little bit and signaling. Nucleic acids, this is the one that you probably haven't heard that terminology, but this includes DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. These are genetic materials. DNA tends to be a storage material. RNA is an intermediate and it can actually have some important functions and does some processes in the cell. DNA just kind of hangs out there and stores the information. RNA is out there doing things in a lot of cases. Particularly, one type of RNA, mRNA, brings the information to the ribosomes, and there the information will get translated into what we call a protein. A protein is a macromolecule made up of amino acids, and a lot of proteins are enzymes. Enzymes are proteins that catalyze reactions. They allow reactions to occur much easier. Some are cell structural components as well, and we have a lot of proteins that are signaling molecules. They can be signals that go out, or they can be the receptors that sit on the outside of the cell and wait for the signaling molecule to come in and basically tell the cell what to do. Again, we'll talk about these in detail in their respective sections and continuing forward in the course. But it's really critical that you know these 4 different types of macromolecules because we're going to come back to them over and over again. These are important concepts in biology. We can't talk about cells without talking about proteins and lipids in the membranes and carbohydrates for energy and nucleic acids for storing the genetic material and creating proteins. So we're going to keep coming back to these over and over again. How do these polymers get formed? Well, there's 2 basic types of reactions that can form these things. First, we have dehydration reaction. Okay, we'll talk about why it's called a dehydration reaction. Then we'll talk about the opposite of that, which is called a hydrolysis reaction. All right, the dehydration reaction is like what we saw earlier. We have 2 glucose molecules here, and we take an OH and an H from those respective molecules. That pops out as water, and a bond is formed between the 2 monomers, linking them together. Because this water has come out, we call this a dehydration reaction. It's being dehydrated. It's kind of a weird terminology. It goes back to the chemistry of this, but basically we're taking these 2 things, taking an OH and an H and sticking them together. Water is released in the process. Now, of course, we're going to have the opposite of this. If we want to break them apart, we have our bonded unit here of our 2 monomers together. If we add water to this, and generally an enzyme is involved in this reaction as well, we can break that bond, and the OH goes back onto this molecule and the H onto this one. We call this hydrolysis because hydro is water and lysis means to break or to separate. So hydrolysis uses water to separate these apart. So if you can build something, you can generally break it. Now let's think about back to our chemistry part of things. Here in the dehydration reaction, we are building a bond. What does that mean energetically? What did we say we can think of bonds as? Well, bonds are stored energy. If we're building one, we got to put energy into it. So dehydration reactions are going to build something that's going to require energy. Okay, let's think about the flip side of that. We already have a bond formed down here. If we break that bond, what's going to happen energetically? We are going to go on and release energy from this in the hydrolysis reaction because we are breaking a bond. A key player in these processes of building and breaking bonds are proteins called enzymes. Enzymes are generally proteins. They're not always proteins, but for our purposes, we can think of them as all being proteins. There's a couple of exceptions. Biology is full of exceptions, but don't worry about it. For our purposes, enzymes are proteins, and they catalyze a reaction. So let's talk about what catalysis is. Enzymes are catalysts. A catalyst is a substance that can speed up the rate of a reaction, but it's not used up in the reaction. Okay, let's examine that. So we have an enzyme here, and it has these two little slots. These slots fit what we call substrates. We have substrate 1 and substrate 2. Substrate 1 and substrate 2 can fuse together. They can bond together to form a product. The enzyme provides them a favorable environment to do this in. So substrate 1 binds with the enzyme and substrate 2 binds with the enzyme. The enzyme puts them in a shape that allows them to easily come together to form the product. And in the process, the enzyme is not used up. That's really critical. Enzymes are super specific to what they can make, or we could do the opposite. There's probably an enzyme that breaks these two things apart, right? They're very specific, though. We'll talk about some examples of this where the molecules are almost identical, but there's an enzyme that breaks down one and not the other. So that's a really critical thing. Enzymes are very specific to what they help bond together. Okay, so that is our first little bit. Then we'll get into the specifics of each of these classes of macromolecules. So our biological macromolecules, they're large biological molecules. We have carbohydrates, lipids, nucleic acids, and proteins in there. Many of these macromolecules are polymers. They're long chains of monomers joined together, generally by covalent bonds. The dehydration synthesis reaction attaches monomers together. That releases water in the process. That generally requires energy to happen, right? Because we're building bonds. The opposite, hydrolysis, that reaction breaks apart polymers. That uses water to do that. And this is generally going to release energy because we are breaking bonds apart. And finally, we talked about enzymes. Enzymes are going to be really important. They're proteins that act as biological catalysts. Catalysts speed up chemical reactions. So enzymes are proteins that speed up biological reactions. All right, we will dive into each of these in the next respective sections.